Welcome students. Last episode we discussed the various enzymes that are involved in the DNA replication. In the continuation of this, in this episode we discussed the DNA replication and how the DNA is replicated in prokaryote and eukaryote. First of all, DNA replication is the process of producing two identical replica from one original DNA molecule. This biological process occurs in all living organisms and is the basis for biological inheritance. DNA is made up of two strands and each strand of original DNA molecule serves as a template for the production of complementary strand, a process referred to as semi-conservative replication. Before the replication, the DNA has two important functions. One is heterocatalytic function, that is when DNA directs the synthesis of chemical molecules other than itself. Example, synthesis of RNA, protein, example. Then such functions of DNA are called heterocatalytic function. Second one is autocatalytic function. The function of DNA which directs the synthesis of DNA itself are called autocatalytic function. Here we are concerned only with autocatalytic function of DNA. Before going to the replication, we have discussed some of the important phenomena which related to the DNA replication. First one is replication fork. The replication fork is a structure that forms within the nucleus during DNA replication. It is created by helicases enzyme which break the hydrogen bond holding the two strands together. The resulting structure has two branching prongs, each one made up of single strand of DNA. These two strands serve as the template for the leading and lagging strand which will be created as DNA polymerase. Matches complementary nucleotides to the template. The template may be properly referred to as the leading strand template and the lagging strand template. The next phenomena is replication bubble. Replication bubble is formed during replication in both eukaryotic and prokaryotic DNA. It is the place where replication occurs actively and also known as replication eye because it looks like an eye of an human. Formation of the replication eye provide the theta-like structure to the circular DNA during replication in prokaryotes. Each replication bubble found to have two replication forks each at the corner of an eye. The next one is Okazaki fragment. Fragment synthesis during lagging strand formation of replication was identified and proved by Regis Okazaki. Hence, by his name, these fragments are called as Okazaki fragments. They are short polynucleotides with 1000 to 2000 base pair in length. These fragments are synthesized by DNA polymerases. Even though they are formed during replication, they are joined to form larger DNA at the completion of replication by the action of DNA ligase. The invention of Okazaki fragment led to the proposal of semi-discontinuous replication concept. Bidirectional replication. DNA replication typically proceeds in both directions simultaneously at any particular replicon. Note that the leading and lagging strand at the two replication forks are on opposite physical strand of the double-strand DNA starting at the origin of replication. This creates two replication forks moving in opposite direction. Bidirectional nature of replication was identified by studying the appearance of nucleotides at the replication forks in connection with the change in the radioactive material intensity. The next one is replicon. Initiation of DNA replication at origin lead to the concept of replicon. A replicon is a DNA molecule or RNA molecule or a region of DNA or RNA that replicates from a single origin of replication. Each replicon has an origin and a terminus. Bacteria and viral chromosomes usually contain a single replicon per chromosomes. In eukaryotes, each chromosome is made up of several replicons. Example, 3500 replicons in 4 chromosomes of Drosophila. The definition of replicons is somewhat confused with mitochondria as they use unidirectional replication with two separate origins. The common method of replication is divided into three types. First one is semi-conservative replication of DNA, then conservative replication of DNA, then dispersive method of DNA replication. Although three types of replication, only semi-conservative method of replication is occurred in prokaryote and eukaryotes. There are various experimental evidence for the semi-conservative DNA replication. First, the experimental evidence for semi-conservative DNA replication in E. coli. The Watson-Crick model of DNA structure and replication suggests that once a replication is initiated, two original polynucleotide strands of the duplex or helix will unwind at least locally so that each strand can serve as a template for a new strand. An immediate prediction follows from this proposal. Both duplex that result from replication should be hybrid in nature. Each contain an old strand derived from the original molecule and a new strand which has been formed during the replication process. Since each of the two double helix or duplex conserves only one of the parent polynucleotide strand, the process is said to be semi-conservative. 4 duplex would result, 2 of which would contain a single strand derived from the original chromosome and 2 of which would contain totally a new DNA strand. Semi-conservative replication is the mechanism by which DNA replicates in cells. The double-strand DNA unwind hydrogen bonds are broken between the two strands by the enzyme DNA helicase. One of the strands is used as a template to form a second complementary strand. The exposed bases on the single-strand DNA complementary base pair to nucleotides. Adenine pairs with thymine and cytosine pair with guanine. Together, the template strand and the complementary strand bond together to form a new double strand of DNA using the enzyme DNA polymerase. One parent double strand of DNA will become two daughter double strand of DNA, each consisting of a strand from the template and the newly formed strand. The new strand of DNA is made in the 5' to 3' direction as the deoxyribonucleotide is added to the 3-hydroxyl end of the chain, which is catalyzed by DNA polymerase. The term semi-conservative refers to the fact that each of the daughter double helix contain one conserved strand from the parent DNA as well as one newly synthesized strand. Conservative type of replication. In conservative replication, both strand of the parent double helix would be conserved and the new DNA molecule would consist of two newly synthesized strands. The next one is dispersive DNA replication, in which the dispersive replication, the replication would involve fragmentation of the parent double helix and the intermixing of pieces of the parent strand with newly synthesized pieces, thereby forming the two new double helix. Iselson and Stock devised an experiment in 1958 to test which of this model correctly represent DNA replication. They grew E. coli for several generations in a medium containing a heavy isotope of nitrogen, that is 15N, that was incorporated into nitrogen bases and eventually into the DNA. This labeled the parent DNA. The E. coli culture was then shifted into medium containing 14 nitrogen and allowed to grow for one generation. The cell was harvested and the DNA was isolated. The DNA was separated by ultracentrifugation, during which the DNA formed bands according to its density. DNA grown in 15 nitrogen would be expected to form a band at the higher density position than they grown in 14 nitrogen. Michelson and Stark noted that after one generation of growth, 14 nitrogen, the single band observed was intermediate in position in between DNA of cells grown exclusively in 15 nitrogen or 14 nitrogen. This suggests either a semi-conservative or dispersive model of replication. Some cells were allowed to grow for one more generation in 14N and spun again. The DNA harvested from cell grow for two generations in 14N formed two bands. One DNA band was its intermediate position between 15N and 14N, and the other corresponded to the band of 14N DNA. The results could only be explained if DNA replicates in a semi-conservative manner. Therefore, the other two models were ruled out. As a result of this experiment, we know that during replication, each of the two strands that make up the double helix serve as a template from which new strands are copied. The new strand will be complementary to the parental or old strand. The resulting DNA molecule have the same sequence and are divided equally into the two daughter cells. Hence, the experiment conducted by Michelson and Stark proved that the DNA has replicated in semi-conservative method. Then the evidence for semi-conservative replication of DNA in eukaryotes. J.H. Taylor and Woods in 1957 provided evidence in support of semi-conservative mode of DNA replication in eukaryotes by using the technique of autoradiography and light microscopy in divided root tip cells of the bean, that is Vicia faba. After incorporation of tritated thymidine, when root tips were transferred to unlabeled culture medium in the first generation of duplication, both chromatids were labeled. In the second cycle of duplication, in each chromosome, one of the two chromatids were found to be labeled. This was interpreted as showing semi-conservative mode of duplication. The next method is semi-discontinuous DNA replication. Various experimental evidences have suggested that DNA synthesis is continuous on one strand, that is 3' to 5' strand called leading strand, and discontinuous on the other strand, that is 5' to 3' strand called lagging strand. Since DNA synthesis always proceed in 5' to 3' direction, so on the lagging strand synthesis takes place discontinuously in pieces called Okazaki fragments. Later on, these pieces were fused with the help of ligase enzyme to form an intact lagging strand. Such a DNA replication where the leading strand is synthesized continuously and the lagging strand is synthesized discontinuously, which is referred to as semi-discontinuous replication. Next one is unidirectional and bidirectional DNA replication. Regarding the direction of replication, DNA replication may be of following two types. First one is bidirectional replication. All known DNA molecules, with only few exceptions, replicate as circle and hence initiate within the helix. In the electron microscope, eukaryotic chromosomes are found to contain multiple expansion replication eyes in contrast to single eye of the prokaryotic DNA. Further, DNA synthesis within a given replication unit eye is initiated from somewhere at the near the midpoint of the unit at a second term as the origin. Prokaryotes contain a solitary origin while eukaryotes have multiple origins for DNA replication. Both ends of the eye are moving and serve as replication forks. Two replication forks are then believed to travel in opposite direction until they reach either end of that unit, the two end points being called termini. A given replication unit may or may not undergo bidirectional synthesis in synchrony with the condigenous unit. In either case, the newly replicated strand in adjacent unit will eventually meet. The strands are then linked perhaps by a DNA ligase to form long continuous dotted DNA strands. Second one is unidirectional replication. In case of this type of DNA replication, one of the two ends of the replication eye remain stationary and the other end serves as the replication fork and moves with replication. An example of unidirectional replication is the replication of mitochondrial DNA by D loop, that is displacement loop in vertebrates. Role of RNA primers in DNA replication. No known DNA polymerase can initiate synthesis of DNA without the availability of the primer RNA strand. So before actual DNA replication start, short DNA oligonucleotide segments called RNA primer or simply the primers have to be synthesized by DNA primer enzyme utilizing ribonucleoside triphosphates. This RNA primer is synthesized by copying a particular base sequence from one DNA strand and differs from a typical RNA molecule in that after the synthesis, the primer remains hydrogen bonded to the DNA template. The primers are about 10 nucleotides long in eukaryotes and they are made at intervals on the lagging strand where they are re-elongated by the DNA polymerase enzyme to being each Okazaki fragment. These RNA primers are later exercised and filled with DNA with the help of DNA repair system in eukaryotes, which is repaired by DNA polymerase 1 in E. coli. coli. In bacteria, two different enzymes are known to synthesize primer RNA oligonucleotides, RNA polymerase on the leading strand and DNA primase on the lagging strand. Replicants. DNA replication in prokaryote and eukaryotes is attained in discrete unit called replicants. The number of replicants may vary in genome from 1 in bacteria and 500 in yeast to several thousand in plants and animals. For example, in E. coli, there is a single replicant with the origin identified as genetic locus ORIC. The origin is adenine and thymine rich, a feature that is related to unwinding of DNA to initiate replication. In E. coli, there are also termination sites, each consists of nearly 23 base pair. The process of termination of DNA replication requires the product of TUS gene, that is TUS protein, or TPI, that is ter binding protein, that is termination binding protein, which recognize termination or termination site. The list of prokaryotic and eukaryotic replicants. In bacteria, number of replicants is 1 with the average length of 4200 kilobase with the fork movement at 5000 base per minute. In yeast, the number of replicants is 500 with the average length of 40 kilobase per with the fork movement of 3600 base per minute. In fruit fly, that is Drosophila melanogaster, number of replicants are 3500 with the average length of 40 kilobase per with the fork movement 2600 base per minute. In toad, that is Xenopus laevius, the number of replicants are 15,000 with the average length of 200 kilobase per with the fork movement 500 base per minute. In mouse, musculus, number of replicants are 25,000 with the average length of 150 kilobase with the fork movement 2200 base per minute. The organism bean, that is Vicia faba, number of replicants are 35,000 with the average length of 300 kilobase per with the fork movement is unknown. The role of replisome and primosome. N.K. Sinha and A.K. Orenberg have suggested that the DNA polymerases, RNA primerases and helicases may be associated with one another to form a multiple enzyme complex, the replisome, that carries out the synthesis of leading and lagging strand in a coordination fashion. Such a complex would be highly processive and assure rapid replication of DNA. Likewise, the protein at the replication fork cooperate to form a replication machine. Example, the primase molecule is linked directly to the DNA helix to form a unit on the lagging strand called a primosome, which moves with the fork synthesis RNA primers as it moves. The next one is mechanism of DNA replication in prokaryotes. In vitro DNA replication has been extensively studied in E. coli and in the phages and plasmids of E. coli. In E. coli, the process of DNA replication involves initiation of DNA replication, elongation of the DNA chain, and the termination of the replication end. First, the initiation of DNA replication, which comprises 3 steps, that is the recognition of the origin site, that is the origin of replication is recognized. Opening of DNA duplex to generate a region of single-strand DNA. Capture of DNA binding protein, that is 5' to 3' helicase, also act as the activator of primase. Thus, DNA A or initiator protein ATP complex binds at 9 base pair inverted repeat regions, that is R1, R2, R3, R4 of origin of replication C of E. coli and promotes opening of the DNA duplex in the region of 3 direct repeats of 13 base pair sequence called 13 mers. The opening occurs from right 13 mer leftwards and requires negatively supercoiled DNA and HU or IHF initiator protein. DNA B, that is helicases, is transferred to expose single-strand DNA and cause unwinding of the DNA in the presence of ATP, single-strand binding protein and DNA gyrase. This results in unwinding of DNA duplex and the replication from origin of replication, that is ORIC, proceeds in both direction, that is bidirectional DNA replication. Single-strand binding protein binding occurs on single-stranded region and 2 DNA B complexes. The next step is elongation of DNA chain. This step requires the presence of the following enzymes and factors: DNA B or helicases, also called mobile promoter, then primase, DNA polymerase holoenzyme, single-strand binding protein, RNase H, which which removes RNA primers, DNA polymerase 1, which is used for filling the gap created due to RNA primers, and DNA ligase, which converts primeless or Okazaki fragments into continuous strand. During initiation to elongation transition, the following events occur. As helicase travels in 5' to 3' direction, it generates a replication fork by opening the DNA duplex. The DNA strand have helicase become a lagging strand, DNA primer associated with DNA B helicase, forming the primosome, which synthesizes multiple primers for lagging strand and a single RNA primer for the leading strand. For the synthesis of lagging strand, DNA polymerase 3 has to work on the same strand to which DNA B helicase is bound, but it travels in opposite direction. DNA B helicase, DNA G primase and DNA polymerase 3 helicase works together in strand elongation. Helicase and DNA polymerase assembly remains processive, that is, they remain tightly bound to the fork and stay bound throughout the reaction. Synthesis of lagging and leading strands takes place by somewhat different methods. It is far more complex for lagging strand than the leading strand. Discontinuous synthesis of lagging strand. Primase is taken up from solution and is activated by helicase to synthesize a RNA primer on the lagging strand. The RNA primers are recognized by DNA polymerase 3 helicase on the lagging strand and are utilized for synthesis of precursors or Okazaki fragments. In fact, each new RNA primer is recognized by gamma subunit of DNA polymerase 3 helicase and loaded with B subunit of the same polymerase. This preloaded B subunit may then capture the core of DNA polymerase 3 helicase, which when it becomes available after finishing its synthesizing job on the preceding Okazaki fragment. On completion of the Okazaki fragments, the RNA primers are exercised by DNA polymerase 1, which then fills the resulting gaps with DNA. After DNA polymerase 1 add to the final deoxyribonucleotides in the gap left by the exercised primer, the enzyme DNA ligase forms the phosphodiester bond that links the free 3' end of the primer replacement of the 5' end of the Okazaki fragment. Continuous synthesis on leading strand. In bidirectional DNA replication, the leading strand is primed once on each of the parental strand. The RNA primer of the leading strand is synthesized by RNA polymerase enzyme. DNA polymerase 3 helicase causes elongation of the leading strand, and finally DNA polymerase 1 and ligase enzymes give final touch to the leading strand, as in case of lagging strand. DNA replication in eukaryotes. Eukaryotic DNA replication requires two different DNA polymerase enzymes, namely DNA polymerase alpha and DNA polymerase delta. DNA polymerase delta synthesizes the DNA on the leading strand, that is continuous synthesis of DNA, whereas DNA polymerase alpha synthesizes the DNA on the lagging strand, that is discontinuous synthesis of DNA. Besides these two enzymes, 6 more factors are involved in eukaryotic DNA replication. First one is T antigen, second one is replication factor A or RFA, also called as RPA or eukaryotic single strand binding protein. The third one is topoisomerase 1, and then fourth one is topoisomerase 2. Fifth one is PCNA, that is proliferating cell nuclear antigen, also called cyclin, and the sixth one is replication factor C or RFC. Various functions of polymerase enzymes. Highly fidelity replicase polymerase alpha, which act as a priming DNA synthesis during replication and repair. The polymerase delta, which is involved in the DNA replication of leading and lagging strand during replication and repair, including base excision repair, nucleotide excision repair. The polymerase epsilon has the function of DNA replication of lagging strand during replication and repair mechanism. The polymerase gamma have the function of mitochondrial DNA replication and repair mechanism. The enzyme high fidelity repair polymerase beta has the function of base excision repair and the double strand base repair mechanism. The polymerase eta, which has involved in the translation DNA synthesis related relatively accurate replication past thymine dimers. The enzyme error-prone repair polymerase zeta has involved in the translation DNA synthesis, that is thymine dimer bypass. The polymerase theta has the repair of DNA interstrand cross-links. The polymerase eta have the function of translational DNA synthesis. The enzyme polymerase eta has the translation DNA synthesis occurred during meiosis. The polymerase kappa translation DNA synthesis, that is deletion and base substitution, then double-strand base repair mechanism with the non-homologous end joining. The polymerase lambda has the translation of DNA synthesis. Polymerase mu has the double-strand basis and repair mechanism with the non-homologous end joining. Polymerase nu, which has the DNA cross-link repair. The enzyme polymerase reverse 1 is a basis site synthesis, that is deoxycytidine transferase activity inserts C across from a nucleotide lacking a base. DNA replication in the eukaryotic involves various steps. Before the onset of DNA synthesis, there is a pre-synthetic stage of 8 to 10 minutes duration for the formation of unbound DNA complex. The steps needs only 3 purified protein, namely T antigen, RFA and topoisomerase 1 and 2. The T antigen, using its DNA binding domain, forms a multi-subunit complex with site 1 and site 2 in the presence of ATP and caused local unwinding. More extensive duplex unwinding occurs due to association of RFA and a topoisomerase with the help of DNA helicase component of T with the help of DNA helicase component of TAGO. Topoisomerase helps in unwinding of DNA by altering topology of DNA at the replication fork. The RFA or single-strand binding protein binds to unbound single-strand DNA. The primer RNA synthesis is performed by primase, which is tightly associated with DNA polymerase alpha. DNA polymerase alpha help in synthesis of an Okazaki fragments in 5' to 3' direction. Replication factor C and cyclin help in stitching of DNA polymerase so that polymerase alpha is replaced by polymerase delta, which then continuously synthesizes DNA on the leading strand. Another Okazaki fragment is then synthesized from the replication fork on the lagging strand by the polymerase alpha primase complex, and this step is repeated again and again till the entire DNA molecule is covered. The RNA primers are removed and the gaps are filled as in prokaryotic DNA replication. 3 DNA polymerases are now known to be involved in eukaryotic DNA replication. DNA polymerase as a might function at both the leading and lagging strand, whereas polymerase E and polymerase delta are involved in elongation of the leading and lagging strand respectively. Models of DNA replication. There are 3 models have been proposed for DNA replication in different organisms. First one is replication fork model. In replication fork model, it occurs both in linear and circular DNA molecule and involves the formation of replication forks which either move in one direction in unidirectional replication or in the both direction in bidirectional replication. The second model is rolling circle model. It occurs during viral DNA replication and DNA replicating in E. coli during mating. According to this model, by some initiation event, a nick is made in the duplex circle and this nick has 3' hydroxyl and 5' phosphate termini. Under the influence of helicase and single-strand binding protein, a replication fork is generated. Synthesis of a primer is unnecessary because of the 3' hydroxyl group, so leading strand synthesis proceed by elongation from this terminus. At the same time, the parental template for lagging strand synthesis is displaced. The polymerase used for this synthesis is polymerase 3. The displaced parental strand is replicated in the usual way by means of precursor fragments. The result of this mode of replication is a circle with a linear branch. It resembles the Greek letter sigma and is called sigma replication or rolling circle replication. These are the various methods of DNA replication and the various models of DNA replication. In comprising this episode, the DNA has replicated in the 3 forms of method: semi-conservative, conservative and dispersive method. methods. Even though three types of methods, semi-conservative method is confirmed by Mission and Stock experiment, which has a single strand from the parent and the single strand is newly synthesized. There are various enzymes which are involved in the replication. In prokaryotes, the replication process is proceed in three steps, that is initiation, elongation and termination of replication. Whereas in eukaryotes, different type of enzymes which are involved in the different functions, and the model of replication is explained in replication fork model and then rolling circle model. These are about the DNA replication, and we concluded this episode. We will discuss the new topic, fidelity of the DNA replication and extra-chromosomal replicons in the next episode. Thank you.